Hello everyone, I am Iqbal Rasool. Today we are going to have another topic that is coronavirus, one of the pandemic disease this time. In this topic we have the following subtopics, that is we have the following questions. First one, what is COVID-19? What are the strains of COVID-19? What are the symptoms of COVID-19? Its causes, risk factors, how transmission occurs, and the diagnosis, structure of the COVID-19, function of spike proteins, the proteins which are the most important in COVID-19, and finally spike proteins, basic vaccines and antiviral therapies. Number first, what is COVID-19? This is a common virus that causes infection in our nose, sinuses or upper throat, but the most coronaviruses are not dangerous. After December 2019 outbreak in China, World Health Organization ने क्या किया, identified SARS CoV-2 as a new type of coronavirus. This outbreak quickly spreads around the world, thus it is known as pandemic. SARS CoV-2 is one of the 7 types of coronaviruses, including the ones that cause severe diseases like Middle East Respiratory Syndrome (MERS) and Sudden Acute Respiratory Syndrome (SARS). The other coronaviruses cause most of the colds that affect us during the year, but are not serious threat to otherwise healthy people. उसके बाद हम स्ट्रेन्स की बात करेंगे। क्या इस सार्स कोविड सेकंड जो वायरस जो है, इसमें कितने स्ट्रेन्स होते हैं ये हम देखेंगे। अगर हम नॉर्मली देखेंगे, नॉर्मल जो वायरस होता है, इट चेंज ऑलवेज, इट चेंज इट्स फॉर्म, म्यूटेट एज इट इन्फेक्ट्स द पीपल। अगर हम चाइना की एक सर्वे के मुताबिक उन्होंने 103 कोविड पेशेंट्स लिए और वहाँ पे आफ्टर performing the experiments they found 2 strains which they named S strain and L strain. लेकिन ये जो S strain जो है ये older one है, लेकिन ये जो L strain जो है ये mostly it is found in early stages of the outbreak. लेकिन ये जो 2 strains है इनमें चाहे L strain या S strain जो 2 strains है, अभी तक ये मालूम नहीं है कि कौन सी स्ट्रेन सबसे ज़्यादा डिजीज़ क्रिएट करता है। लेकिन इस पे आफ्टर सर्वे के मुताबिक, आफ्टर एक्सपेरिमेंटली व्हेन ये प्रूव होगा तो उसके बाद हम बताएंगे कि एल स्ट्रेन सबसे ज़्यादा हार्मफुल है, सबसे ज़्यादा डिजीज़ क्रिएटिंग है, या एल स्ट्रेन सबसे ज़्यादा डिजीज़ क्रिएटिंग है। उसके बाद जो सिम्टम्स ऑफ कोविड-19 जो है, हमने यहाँ पे फ्यू सिम्टम्स लिखे हैं, बहुत सारे सिम्टम्स है। पहला वन है फीवर, second coughing, shortness of breath, body aches, headache, loss of smell or taste, sore throat, etc. What are the causes of coronavirus? अभी तक जो काज कोरोना वायरस का है वो अभी तक मालूम नहीं है, लेकिन यहाँ पे बहुत सारे वायरसेस है, इस टाइप के वायरस बहुत सारे हैं जो मौजूद होते हैं इन एनिमल्स, आल्सो इन केस ऑफ ह्यूमन्स। इनके सब एनिमल्स वहाँ पे हम एग्जांपल यहाँ पे हमने दी है, डेट इज बैट, कैमल्स, कैटल एंड कैट्स। लेकिन ये जो सार्स CoV-2 जो है डेट काजेज COVID-19, द वायरस डेट इज वायरस SARS CoV-2 which causes the COVID-19 is similar to that of the virus of which causes MERS, Middle East Respiratory Syndrome, and SARS, Severe Acute Respiratory Syndrome। लेकिन ये जो वायरस MERS और SARS जो इनका जो वायरस है वो उसका ऑरिजिन है बैटस, लेकिन यहाँ पे जो ऑरिजिन COVID-19 का ऑरिजिन ऑफ द वायरस SARS CoV-2 जो है उसका ऑरिजिन अभी तक मालूम नहीं है। but the researchers are finding the origin of the SARS-CoV। उसके बाद जो कोरोना वायरस के रिस्क फैक्टर्स हैं, अभी तक हम उस एरिया में हैं जहाँ पे कोरोना वायरस जो है इट इज़ नॉट स्प्रेडिंग एज फास्ट एज इट स्प्रेड्स इन द अदर कंट्रीज। लेकिन जो पर्सन हु हैव ट्रैवल्ड फ्रॉम द एरिया वेयर इट इज स्प्रेडिंग लाइक चाइना, लाइक ईरान, लाइक अमेरिका, like Italy, etc. When it comes in contact with the healthy person, when it comes in contact with the normal person, the normal person has low risk of infection. But the people who are about over the age of 65 are most likely to get a serious illness. Why they get the more serious effect, why they get more serious illness, it is almost in 65 when the people who are 65 over the 65 age, also the children that is infants, they get more serious effects. Why? Because their immune system is not strong, because their immune system in case of children it is not boosted by the any type of the viral vaccine, as well as in case of older people the immune system gets weak, the immune system is weak, so they do not bear the vaccine, so they do not bear the effect of the virus that causes the COVID-19। यहाँ पे कुछ कंडीशंस है, यहाँ पे जो किन किन पर्सनस में हो सकता है, चाहे वो मेल हो चाहे वो फीमेल है, लेकिन किन किन पर्सनस में हो सकता है जिनमें कोई मेडिकल कंडीशन हो। कौन कौन सी मेडिकल कंडीशन होगी? अगर किसी आदमी में, किसी इंसान में हाई ब्लड प्रेशर होगा तो उसमें भी कोविड-19 के चांसेस ज़्यादा है। अगर किसी इंसान में हार्ट डिजीज है, कोविड-19 के चांसेस ज़्यादा है। सिमिलरली लंग डिजीज, एजमा, किडनी डिजीजेस that need dialysis, obesity, diabetes, कैंसर ट्रीटमेंट, स्पेशली यहाँ पे कौन सा ट्रीटमेंट होता है, यहाँ पे केमोथेरेपी का जो ट्रीटमेंट है, लिवर डिजीज एंड स्मोकर्स, मोस्टली सिगरेट स्मोकर्स। ये जो यहाँ पे जो मेडिकल कंडीशंस है, ये हम जो मैंने आपको बताए हैं, दीज आर द कंडीशंस विच आर रेस्पॉन्सिबल फॉर इन दीज पर्सनस द कोविड-19। इफ द कोविड-19 जो होता है, जो वायरस SARS-CoV-2 का इफेक्ट इन पर्सनस में जिनमें ये मेडिकल कंडीशंस होंगी, इनमें इसका इफेक्ट ज़्यादा होता है, या ये रिस्क फैक्टर से दीज पर्सनस आर ऑन द बॉर्डर लाइन ऑफ द कोविड-19। उसके बाद जो नेक्स्ट स्लाइड है, इसमें क्या है, वो कोरोना वायरस ट्रांसमिशन कैसे हो सकता है, कैसे होती है, और हाउ डज द कोरोना वायरस स्प्रेड। ये जो SARS-CoV-2 जो वायरस है, कोविड-19 काजिंग वायरस, it transmits mainly or it spreads mainly from person to person। लेकिन मोस्ट ऑफ द टाइम अगर हम बात करेंगे, अगर हम कोई भी सिक जो पर्सन है, जो कोविड पेशेंट है, व्हेन इट स्नीजेस और कफ्स, देन then the droplets come out through his nose or from the mouth, and they reach up to the 6 feet away। And if accidentally or if we are present in front of the person, if in front of the sick person, then we inhale these droplets, then these droplets reach in our gastrointestinal tract or respiratory tract, and we also get sick। But if some persons do not show sign and symptoms due to the fact they have strong immune system, but the only persons whose immune system is weak and the persons whose age is more than 65 years and the children, they show the effect because the immune system in these persons is weak। We can also get the virus from touching the surface or the object on which this virus particle is present। अगर हम क्या करते हैं, जब हम टच करते हैं किसी भी सब्सटेंस के से जहाँ पे ये पार्टिकल है बाय हैंड, एंड इफ वी टेक दिस हैंड, और इस ये जो हैंड जो है उसके बाद हम टच करेंगे अपने नोज के साथ, आईज के साथ और माउथ के साथ, देन दिस वायरस पार्टिकल एंटर्स इनटू आवर रेस्पिरेटरी ट्रैक्ट और गैस्ट्रोइंटेस्टाइनल ट्रैक्ट। फाइनली वी आर आल्सो सिक, देन वी आल्सो बिकम द कोविड-19 पेशेंट्स। और एक स्टडी के हिसाब से, अगर हम एक स्टडी के हिसाब से देखेंगे हाउ मच टाइम, ये जो टाइम पीरियड है कोविड, ये जो SARS-CoV-2 जो वायरस जो है कितना टाइम ले सकता है, अगर ये किसी भी सब्सटेंस पे होगा तो इसका टाइम पीरियड कितना होता है, कितनी देर तक ये वहां पे रह सकता है। यहाँ पर हमने कुछ एग्जांपल्स दी है। फॉर एग्जाम्पल अगर कापर की बात करेंगे वहाँ पे 4 आवर्स ही रह सकता है। कार्डबोर्ड की बात करेंगे वहाँ पे अप टू 24 आवर्स रह सकता है। प्लास्टिक और स्टील स्टील की बात करेंगे वहाँ पे 2 to 3 days रह सकता है। This is the reason. This is the reason why it is important to disinfect infected surfaces to get rid of the virus. लेकिन some dogs and as well as cats have tested positive for the virus for the COVID-19. यहाँ पे जो डॉग्स और कैट जो टेस्टेड पॉजिटिव है दे शो सम साइंस ऑफ द इलनेस आल्सो। इन एडिशन टू दैट आफ्टर द डॉग जो वायरस पॉजिटिव है जो कोविड-19 पॉजिटिव है और कैट दैट इज नाउ कोविड-19 पॉजिटिव, यहाँ पे कोई भी एविडेंस अभी तक नहीं है कि इट नाउ ट्रांसमिटेड फ्रॉम द कैट और इट नाउ ट्रांसमिटेड फ्रॉम द डॉग व्हिच इज नाउ कोविड-19 patient which is now COVID-19 positive, sorry। उसके बाद जो नेक्स्ट हमारी स्लाइड है डेट इज कोरोना वायरस डायग्नोसिस। इसके जो डायग्नोसिस है फ्यू सिम्टम्स यहाँ पे हमने दिखाए हैं कौन कौन सा जो साइन है। डेट इज फीवर हो सकता है 100 फॉरेनहाइट और हायर, कफ हो सकता है और ट्रबल इन ब्रीथिंग। और अगर हम ये हाउ टू डिटेक्ट दिस कोरोना वायरस, उसके लिए हमारे पास एक टेस्ट है डेट इज स्वैब टेस्ट इज मोस्ट कॉमन मेथड फॉर द डिटेक्शन ऑफ द कोविड-19। But in some areas there are also tests that is rapid test that results in a little as in 15 minutes। लेकिन ये जो कुछ टेस्ट है फॉर एग्जांपल जो हमने स्वैब टेस्ट बताया या डेट इज रैपिड टेस्ट बताया, इन एडिशन टू दैट देयर इज एंटीबॉडी टेस्ट। दिस swab test only tells us whether the virus is present in our body at that time, but the antibody test shows whether you have ever been exposed to virus. This is one of the importance of the antibody test. Now the structure of the COVID-19 virus. This virus, this SARS-CoV-2 virus, it contains mostly the spike proteins. The short name of the spike protein is S protein, and this spike protein is a large type of first transmembrane protein, ranging from 1160 amino acids for avian infectious bronchitis virus, that is IBV, and up to 1400 amino acids for feline coronavirus, that is FCOV. In addition to this protein, that means this spike protein is highly glycosylated and it contains about 21 to 35 N glycosylation sites. And these spike proteins assemble into the trimers on the viral surface to form the distinctive corona or crown-like appearance. उसके बाद जो एक्टोडोमेन ऑफ द कोरोना वायरस जो है, जो एक्टोडोमेन ऑफ द कोरोना वायरस स्पाइक प्रोटीन्स जो है, दे हैव द सेम ऑर्गेनाइजेशन इन 2 डोमेन्स। कौन कौन डोमेन है? नंबर 1 डोमेन एन टर्मिनल डोमेन विच इज नेम्ड एज S1। दिस डोमेन इज रेस्पॉन्सिबल फॉर रिसेप्टर बाइंडिंग। एंड द नेक्स्ट डोमेन इज C टर्मिनल डोमेन विच इज नेम्ड एज S2 डोमेन, and this domain is responsible for the fusion। और ये जो कोरोना वायरस की जो डाइवर्सिटी है, ये डाइवर्सिटी ड्यू टू variable spike proteins, the structure of the variable spike proteins which are present in the coronavirus. And this variability evolved into forms differing in their receptor interactions and their response to various environmental triggers of the virus cell membrane fusion. It has been noted that this COVID-19 virus, it can cause the infection in case of respiratory tract। सिंपली ये यहाँ पे जो ह्यूमन रेस्पिरेटरी ट्रैक्ट में जो epithelial सेल्स जो होते हैं वहाँ पे ये इंटरेक्शन दिखाता है विथ ह्यूमन ACE2 receptors which are present on the epithelial cells वहाँ पे। उसके बाद indeed the recombinant spike proteins can also bind with the recombinant ACE2 protein। लेकिन यहाँ पे जो डिस्टिंक्शन जो यहाँ पे है, जो बहुत सारे जो स्पाइक प्रोटीन जो होते हैं उनमें क्या फर्क है विथ रेस्पेक्ट टू द कोरोना वायरस, यहाँ पे ये है डिफरेंस है कि वेदर इट इज क्लेव्ड और नॉट। during the assembly and exocytosis of the virus. With some exceptions, अगर हम exceptions के बाद most alpha coronaviruses and beta coronaviruses, that is SARS-CoV, the virus harbor a spike protein that is uncleaved. लेकिन in some cases, that is beta and all gamma coronaviruses, the protein is found cleaved between the S1 and S2 domain, typically by furan. This furan is Golgi resident host protease. Interestingly, within the beta coronavirus, mouse hepatitis virus, that is MHV species, different strains such as MHV2 and MHVA59 display different cleavage requirements. This has important consequences on their fusogenicity. On the next slide you will show वहाँ पे diagram है वहाँ पे. Now spike protein structure. This coronavirus spike protein is a class first fusion protein. The formation of an alpha helical coiled coil structure is the characteristic feature of this class of fusion protein, which contains in their C-terminal part regions predicted to have an alpha helical secondary structure and to form coils. The S2 subunit is the most conserved region of the protein, whereas the S1 subunit diverges in sequence, even among species of a single coronavirus. The S1 contains two subdomains. First one, N-terminal domain, short form NTD, and a C-terminal domain, short form CTD. Both are able to function as receptor binding domains, RBDs, and bind a variety of proteins and sugars. This coronavirus spike proteins contain two heptide repeats in their S2 domain. This is a typical feature of a class 1 viral fusion protein. This heptide repeats comprise of a repetitive heptide peptide A, B, C, D, E, F, and G, with A and B being hydrophobic residues, which is characteristic of the formation of the coiled coil that participate in the fusion process. For SARS-CoV and MHV, the post-fusion structure of the HR has been solved. They form the characteristic 6 helix bundle. The functional role of MHV and SARS-CoV HR was confirmed by mutating key residues and by inhibition experiments using HR2 peptides. This is the diagram of SARS-CoV spike protein. The spike protein ectodomain consists of S1 and S2, as here it is shown in the diagram. The S1 domain contains the receptor binding domain and is responsible for recognition and binding to the host cell receptor. And the S2 domain is responsible for fusion, contains the putative fusion peptide, which is colored in below, and the heptide repeat HR1, which is colored in orange, and HR2, which is colored in brown. The transmembrane domain is represented in purple, and the cleavage sites are indicated with arrows. Now, what is the function of spike proteins? The COVs are widely distributed in nature, and their zoonotic transmission into human populations can cause epidemic diseases. After entering into the respiratory or gastrointestinal tract, these viruses establish themselves by entering and infecting lumenal macrophages, and epithelial cells. The cell entry programs for these viruses are ushered by the viral spike, that is S proteins, that bind cellular receptors and also mediate virus cell membrane fusion. Take SARS-CoV for an example. The spike proteins S protein of SARS CoV virus has pivotal role in viral infection and pathogenesis. The S1 domain recognizes and binds to host receptors, and subsequent conformational changes in S2 domain facilitate fusion between the viral envelope and host cell membrane. Models depicting the S-mediated membrane fusion event have extending from knowledge of S protein structures and functions. In part, these models are deemed responsible because the post-fusion 6HB conformations in SARS and MHVS proteins are so strongly similar to post-fusion forms influenza HA2, paramyxovirus F2, Ebola virus GP2, and HIV GP41. In analogy to these more widely stated and well understood viral fusion proteins, the COVS mediated membrane fusion process is generally shown in the diagram. This is the schematic COV spike protein mediated membrane fusion. This represents several steps of S protein conformational change that may take place during the membrane fusion. In first step, receptor binding pH reduction and or S protein proteolysis induce dissociation of S1 from S2. This setup is documented to this setup is documented for some MHVs. In the next step, the fusion peptide Fp is intercalated into the host cell membrane. This is the fusion intermediate stage. In the third stage, the part of the S protein nearest to the virus membrane refolds onto a heptade repeat 1, that is HR1, core to form the 6 helix bundle, that is 6HB, which is the final post-fusion configuration of the S2 protein. Now finally we come across the spike protein based vaccines and antiviral therapies. The role of spike proteins in receptor binding and membrane fusion indicates that vaccines based on the spike protein could induce antibodies to block virus binding and fusion or neutralize virus infection. Among all the structural proteins of SARS-CoV, spike protein is the main antigenic component that is responsible for hosting immune responses, neutralizing antibodies, and or protective immunity against the virus infection. Spike proteins have therefore been selected as an important target for coronavirus vaccine and antiviral development. Now here are some vaccines which are against the COVID-19. If the vaccines are full-length S proteins, these full-length S protein induces effective neutralizing antibody and T cell responses as well as protective immunity. But the disadvantage of this vaccine is that it might induce harmful immune response that cause liver damage. Next one is DNA based. The advantages of this DNA based vaccine is it is easier to design, induce immunoglobulin G, neutralizing antibody and T cell responses, and or protective immunity. But the disadvantage of this DNA based vaccine is that It might have low efficiency in humans. Repeated doses may cause toxicity. After that, there is another, that is viral vector-based vaccine. This induces the advantage of this vaccine is that it induces neutralizing antibody responses, protective immunity, and or T cell responses. But the disadvantage is that it might induce ADE effect, possibly present or pre-existing immunity. Another one is recombinant S protein based vaccine. Advantages of this vaccine is that it induces high neutralizing antibody responses and protective immunity. But the disadvantage is that mainly humoral responses need repeated doses and adjuvants. After that, the next vaccine is RBD. This induces the highly potent neutralizing antibody and T cell responses and protective immunity. But the disadvantage of this RBD is not yet identified. Another one is viral vector-based vaccine. This induces the neutralizing antibody responses, protective immunity, and or T cell responses. But the disadvantage of this is that it induces possible genomic integration of foreign DNA, viral vector instability. There are other vaccines also, but these are some vaccines which are yet to be developed or which have the advantages as well as disadvantages. I have told you that. After that, there are some therapies which are induced against which are applied against the COVID-19. Number one is peptide. This is one of the therapy. The advantage of this therapy is that it inhibits virus infection by preventing S protein mediator receptor binding and blocking viral fusion and entry. But the disadvantage is that it has a low antiviral potency. Another therapy is that RBD ACE2 blockers. This blocks RBD ACE2 binding and S protein mediated infection, but the disadvantage is yet to be identified. After that, there is a next that is S cleavage inhibitors. These might interfere with S cleavage, but the disadvantage of this another is also not identified. Next one is fusion core blockers. Easy to design, inhibits virus infection with high specificity, but the disadvantage of this fusion core blocker is also yet to be identified. Another one is neutralizing human antibodies. This inhibits the virus entry, neutralizes virus infection, induces cross protection, and reduces diseases severity and viral burden. More suitable for development as human immunotherapies. But the disadvantages of this neutralizing human antibodies are also yet to be identified. And there are also some others like carbon small compounds. These are oral bioavailability but low antiviral potency. Another one is that is small and interfering RNAs. These reduce the virus replication and are silences as gene expression, but they have also low antiviral potency and limited usefulness. There are also some other therapies which are involved in against the human coronavirus. This was about the COVID-19. This was information about COVID-19. Next to meet in the next session. Take care. Allah Hafiz.